Welcome back. This is the second part of Lecture 10 for the Computational Neuroscience course, System 552, Biology 487, taught at the University of Waterloo in the winter of 2021. This is where we're going to cover a quick overview of high-level visual processing to carry on from the low and intermediate level parts that we just did. All right. So this is going to be sort of very, very high level. We're sort of, after all this, low level intermediate processing of the raw image features that are coming in, the color, the polarity lines, lines of different orientations being extracted, surfaces, all those sorts of things at the intermediate visual processing. The point of all of that seems to be to try to do something about object recognition and using this image input as something useful for behavior, is I guess the right way to put it. And we will talk mostly about the ventral stream, mostly about the pathway that is doing object recognition. But then in the end, we'll sort of cover a little bit of things that are more about the dorsal stream, the one that's more about using what you've observed to control sensory motor actions. And in particular, the thing we'll look at is using what you're seeing to direct your attention to different places. So we'll see how that all goes. The reading is Kendall et al. Chapter 28 and 29 if you feel like reading a little bit of extra. Cool. So, all right, let's start with the most typical, sort of most well understood thing that vision is for is recognizing objects. And this seems to be something that is we get our raw features coming in from the retina into V1, and then we build up more complex features and build up more complex features. And eventually, once we get up to inferior temporal cortex, or IT, as we're sort of coming this way along the brain, we're getting closer to the parts of the brain that are more about audition and language. And so we're getting closer to something about labeling these images. We're also tying in, as we're getting closer to this pathway, it's not just about labeling the images, we're also going to be getting, starts getting closer to where you want to actually feed data over to things like the amygdala, which is, we talked about, was recognizing emotional salience of our stimuli. Can also start getting this also be getting closer to hippocampus, so we can start using what we're seeing to either store things in long-term memory or recall things from long-term memory. That's the sort of general idea of why you might want to have something that takes in an image and categorizes it somehow. As usual with high-level cognitive things, one of the fun things to start looking at is, hey, what happens when it goes wrong? And in this particular pathway, a couple different interesting things can go wrong. Well, many interesting things, but two particular ones are, here on the left, we're showing an example of aperceptive agnosia, which is generally damage to this part right here. And the symptoms there is I can show you a picture of a dark circle here and ask you to draw it, and they do this, and they go, I don't know, some squiggles. But you ask them exactly what it is, and they say, oh, it's a circle. The difficulty is that they're having difficulty representing the picture itself somehow. They can recognize it fine, but they don't seem to be able to represent or be able to use that representation of the circle. Same thing, I show them a square, they can tell it's a square. Their drawing does not look like a square. In contrast, we've got a slightly different part of this pathway a little bit later, which is called associative agnosia, where I can show them a picture, and here we're doing a nice complicated picture. Here's I guess it's a tea bag, and you ask them to draw it, and they draw it fine. You ask them to identify it, nothing. I don't know, they're just perfectly good at representing what they're seeing, not good at mapping it onto terms. Okay. So though that seems to be the sort of task that seems to be involved when I damage things way up here at this later part of the of this particular image pathway that is going to be recognizing objects. Cool, weird. How does that work? It just seems strange. And one of the things that I think really gets me excited seeing these sorts of things is I have no idea how I would build a system that when it got damaged, this is the sort of thing that happens. Like that's just weird. And to me, that's the sort of fun data. If I'm trying as a computational neuroscientist to try to build a model of how the brain works, my model, when it's damaged, really should show these same sorts of effects. So this sort of data gives great evidence of what you want your eventual computational model to explain. And it's an incredibly hard puzzle to try to figure out how you're going to go ahead and build a model that would end up exhibiting these sorts of symptoms. But gives us very clear targets. What other things have people, fine, gross damage to this area or large-scale damage to this area seems to have these very strong effects. What else can we study about? And then the other place you get lots of data about neuroscience is animal studies and going ahead and actually recording from the brain when there's different stimuli being presented. And so here is an example. This was done in monkeys, where we're recording from part of the inferior temporal cortex. So IT. This is the sort of a later stage in the visual hierarchy. And what we're showing here across the bottom. So the graph is showing the activity of a particular neuron, and some stimulus is being shown for some period of time. That's this little window down here. So what we're seeing initially is some sort of background activity of the neuron. The neuron's just sitting there, you're not looking at anything. And then you show the monkey a picture of a monkey, and this neuron starts firing a lot. You show the monkey a sort of a similarly structured image, but that is definitely not a monkey, and the neuron doesn't fire at all. Showed a different monkey. It fires a slightly different pattern. Monkey with the mouth sort of cut off. So we're missing parts of the image here. Still is active. Missing other parts of the image. Still active, but differently. Whether or not there's color doesn't, also affects things. So certainly this neuron is firing slightly differently for these different stimuli. So something different is being represented, but this neuron is still very preferentially triggered by faces. Turns out also triggered by human faces, but then this particular neuron is, say, not triggered by, say, pictures of hands. Whereas you can go find other neurons in the similar sort of area that end up being that, yes, okay, these ones are preferentially start firing whenever you show a hand. So that's sort of how we can start getting the sense that, yes, this is what this part of the brain is doing. It's identifying these large-scale sort of objects. And then things can get a little bit more specific. We can start looking at, oh, hey, look, here's ones that maybe seem to be a little bit more sensitive for particular emotional expressions, or faces seen front on or from the side. There's going to be neurons that are doing all these sorts of different things. And this is why we're sort of can think of these neurons as these are picking up very, very complicated features. These are very complicated features, but we can still kind of think of them as features being extracted from the image. Just in a very complicated way. Yeah. So that's one aspect, that they're picking up very, very complicated features. Another aspect that seems to be true about neurons at this sort of much later part of the visual hierarchy is they're pretty insensitive to where the object is. So it doesn't really, it's not like, okay, there's a neurons that detect faces at this particular location in space. No. This is more like, okay, I'm detecting faces no matter where they are. Some difference, and that's exactly, so this is presenting faces at different locations. Fine. But this same neuron seems to be working pretty well there. Also, it's going to be relatively constant across different sizes of faces. And even sort of, is it, here's the same shape, but sort of white on a black background versus black on a white background. Okay. So this sort of invariance, these are things that the features end up being not sensitive to. And what's interesting is these sorts of invariants, this seems really useful. It seems it would be really good to have a feature that just says, is there a face or not, regardless of where it is in the visual field. But it's really tricky to build a feature that does that. Like if we're thinking about building features the way that we did with neural networks, or that we do with neural networks, which is, okay, well, I've got some inputs, and I'm going to do some weighted sum of my inputs, and then apply some sort of nonlinearity. Certainly, we can't do that with just one layer of such network, of such an input. If we have enough of these neurons, we can do that, but figuring out exactly how to wire up a network to extract that sort of feature is pretty tricky, and indeed that's what a lot of deep neural network sort of research ends up being, is how do you train up a network such that it can identify these sorts of things, identify faces wherever they are in the visual field. The brain seems to do it pretty well. The brain seems to do it in fewer layers than a lot of the deep neural networks that we're talking about, or that people use, but that could be because the brain is making use of a lot of these weird lateral connection type things that we were talking about in the previous lecture about the intermediate layers. Cool. So we know, we know that the brain does this. Exactly how it does this is a little unclear. Especially since it gets even more complicated than that. So you start getting things that are invariant across, all right, it's the same objects, but seen, different images of the same object, or different orientations of the same object, or like different characterizations, like all of these are a horse, but these are very different sensory stimuli. And then it's going to be even more weird that it can start doing things like, well, it's not just images that can start triggering these neurons. It can start being, oh, okay, hold on. We're also able at this point to start getting input from other sensory modalities. So the sound of a horse, or the smell of hay, all of these sorts of things can also start triggering these neurons. So this is why we started thinking of these neurons as more representing the object, the essence of the object, rather than just a visual representation of the object. Again, it's a situation where humans do this really, really well. It's trickier to get AI neural network type things to make this sort of generalization. Okay. How do we study this sort of stuff? There's some cool experiments out there. Here's one, training up a bunch of monkeys. We're recording data from monkeys. And the stimuli that we're giving them, we have a bunch of sort of 3D generated images of cats and images of dogs. And then because these are 3D generated, you can morph between them. And so what we've got here is a gradual morphing from cat to dog. Right, so the features are, like the low-level visual features are pretty darn similar across these things. Right? But this one here, hopefully you can see it in the image, is pretty strongly a cat, and this one here is pretty strongly a dog. And if someone was going to ask you exactly how you made that distinction, it'd be a little tricky to say exactly what the difference is. But then what we can do is put a monkey in a situation where, okay, we're going to show it an image. It's going to have two buttons to push, one button if it's a cat, one button if it's a dog. We're going to give it a reward if it pushes the right button, so it's motivated to push the right button. And then we start showing it, we get it working fine on these sorts of extreme cases. It knows what it's doing, and then we start showing it these intermediate pictures. And then there's two things we can measure. We can measure, well, what button do they push? And then we can also measure neural activity in this sort of region. And the fun thing that we end up finding is, yes, we're going to, while we're keeping track of what this object is, you get these neurons that are, so this is the same neuron. All these different lines are the same neuron, and we're plotting here sort of the average activity given different inputs. And for the ones that are cat, that are more like the cat, the neural activity is very different than the ones where it's more like a dog, where we have this other neural activity. Not only that, but it also turns out that if you look at the individual choices that the monkey is making, if the neuron happens to be firing more, because the monkey is not 100% perfect at distinguishing these 60% cats from 60% dog pictures, but the situations where the monkey goes, oh no, I think that's a dog, those are, the neural activity is more like these situations here, where it's consistently saying it's a dog. So that's partly, that gives us a little bit of evidence that that whatever it is that's driving the behavior, the decisions of the monkey, is based on whatever these neurons are doing. That's sort of the core idea there. So again, kind of nice evidence that we are extracting some sort of very high-level information about the image. And that this information, we can look at individual features being pulled out and sort of say, oh, okay, that there are individual neural activity, and that individual neurons seem to be tied to particular high-level features, like is it a dog, is it a cat? I will definitely say that it's pretty rare to find a neuron that is only sensitive to one high-level feature. You will often find neurons that, so this particular neuron is sensitive to dog versus cat, but it might also be that this neuron also responds to airplanes and helicopters and mice, some other weird combination of things. So that's sort of more highlighting that it's not one neuron on its own that represents dogs versus cats, which is good, because if that neuron died, then you wouldn't be able to recognize dogs, and that'd be kind of weird. It is one feature being extracted, but the actual sort of your overall labeling of the whole system is going to involve many, many features. So if you have many features, then you can do more categorization, do better categorization, just like we saw in the sort of perceptron and artificial neural network cases. As you have more features, you can do more things. Cool. So that's a big aspect of okay, recognizing objects, fine like that. There's a fun part of this area, of this brain area, called the fusiform face area. It's been studied for a long time. It's part of IT, or right in there. It's one of the better studied areas, just because it seems to have this fascinating connection to faces in particular, and faces seem to be pretty important for at least primate and lots of other mammals. And this particular video I'm going to show here, so this is a brain area where we seem to have neurons that are very preferentially associated with faces. Okay, so the features that seem to be picked out are faces. But then there's this strong question about, well, what does this brain area actually do? I mean, we know that these features are there. We know that there's some neurons that are picking out, is this a face or not? But how is the brain using that information? And this particular video is absolutely fascinating. What we're going to be doing is we're going to...
by asking how electrical stimulation of that region affects the patient's percept.
Just what I said there.
Here is our movie.
So,
and importantly, this patient...
Oh, and sorry, before I continue there, this researcher is Nancy Kanwisher from MIT, and lots of colleagues. This whole video that is linked there is a fascinating exploration of both the fusiform face area, but also in general, how you can use brain scanning information to really understand what the functionality of different parts of the brain are. But this, I think, is a really striking example of the fusiform face area, and what we're going to do is try stimulating it and see what happens.
Doesn't know about the fusiform face area, and he doesn't know where his electrodes are. You can see him getting stimulated right there. You can watch.
言葉であれするですね。もう一回やってもう一回。One more time. One more time. この部分だけなんかアニメでできそうな感じのキャラクターの顔に変わったんです。はい。
So I'll continue that a little bit, but so I don't know how visible that text was, but yeah, stimulate this brain area while this patient is looking at a person, and what he reports is that his eyes and nose turn into an anime character. That's really weird. Now we're going to do another example.
So that shows the causal role of that region in face perception. The question here is, what is its causal role in object recognition? Does it affect the percept of an object?
No change. 最初の1秒ぐらい、始まって1秒ぐらいがなんか横になんか目口って感じでちょっと一瞬見えたんですけど、これ何かなと思ってたら気が付いたらもうこの箱にしか見えてないっていうか。なんですかまたさっきみたいに横にその目と口って感じでちょっとアイス。
This is a kanji character. It's foreshortened, so it's hard to see.
More, please. One more time. やっぱり顔に見えました。目口らへんって感じ。
Okay, you get the idea.
Okay, that's just fun. So you stimulate this area, and it's not just that the features. So yes, this area has neurons whose features are extracting. Is there a face or not? But if I stimulate this area, and by stimulating this area, I'm changing those feature values because I'm causing these neurons to fire or not fire, or change their pattern of activity. And whatever it is the rest of the brain is doing with this information, it's causing this person to start hallucinating eyes and mouths and faces where there are no faces. So it not only disrupts, if you are looking at a face, it disrupts the exact details of that face, which somewhat makes sense because we're just sort of stimulating this whole area. So it's just like changing the feature values of a whole bunch of neurons almost at random. But it's still something about faces. And if I do that when you're looking at a standard object, we start hallucinating eyes and mouths. And that's just kind of nifty. Anyway, the whole video is definitely worth watching if you're interested in more stuff like that.
Objects by
cool. What else do we want to talk about? This sort of these neurons and this sort of activity also seems to be involved in a little bit of working memory. So it's not just, hey, am I seeing this object right now? But am I remembering this object? So in this sort of task, I'm shown an object, and then the object goes away, and there's some waiting period. And then I'm shown 4 objects, and I have to go look at the object that was the same as the one I was seeing before. So during this delay period, you have to remember what you looked at before. If you go ahead and take a look at these neurons, they're actually sensitive. They activate during that whole delay period. So it's not just when the cue is present, but during that whole delay period. And then, so that seems to be some evidence that the feature is, we need a little bit, we're doing something a little bit fancier than just sort of, hey, yes, here's some input image, here's your input image, we're doing a bunch of image processing. We also need to be able to store this information over time. Cool. So again, something that normal features in artificial neural networks don't do. All right. So other sorts of things that so that was all about sort of object recognition and just sort of, hey, is there an object? Can I use this information? There's also a lot of visual information that is wanting to be used more about tying this visual information to sensory motor tasks. So it's not, I don't care about what the object is, but I might care about where things are in order to move my eyes toward them or respond differently. And one big particular thing that might be really useful for a visual system to do is if there's something interesting that I'm seeing, but I'm not directly looking at it, I might want to move my eyes to point at that. Because as we covered in the low-level visual processing things, you get a lot more detail about the thing you're directly looking at than anything on the periphery of where your eye is. So something needs to control moving your eyes around. And that really should be driven by vision itself. So this is called attention, or and the whole idea is going to be that we've got a couple different types of attention. So there's involuntary attention, where you're really not very much in control of it. So it's much more stimulus driven, sometimes bottom up. And this is more the idea of I'm looking at a screen and all of a sudden a dot appears. My eyes jump to that dot really quickly without me controlling it. It's just going to happen. Okay, so and it's also going to be the case that even if it's not just something suddenly appearing, but even if it's here's a more complex shape across a background of really simple shapes, I might go ahead and look at that more. So that's one aspect. And then there's also voluntary attention, where you're being told, oh, hey, look, pay attention to this side of the screen, or something like that. And that can either be in a situation where you can actually tell people, okay, well, stuff is going to happen on the left side of the screen, so look over there more. But then you can also do it in situations where you're forcing people to look directly ahead, but just ask them to pay attention to the left. So it can be hard for them to control their eye movements in those sorts of situations. In any case, if you can get your attention or your eye movements to particular locations, there's lots of evidence that's going to decrease reaction time, so you're going to respond faster. You're also going to be able to pick out smaller changes in details better, just because you're getting more data from the correct area, so you can be a little bit more accurate. So better contrast detection. And so we need some sort of system that are going to, that's going to sort of control this. And one cool thing that seems to work pretty well, because we can do things like show people a picture, like this painting here in the middle, and then just track where their eyes go. And you get these really clean stereotyped things that even if you don't tell them anything of what to do with this image, you get some very clear things that people look at people's faces. That's what we're seeing here. These blue dots is the path of the eye movement. And also seem to be looking at fairly sort of important points, feet here, so identifying this person in this particular case. So this sort of pattern ends up being pretty consistent. And very unconscious. So again, it's not a situation where we're asking someone to do anything in this picture. They're just saying, look at the picture. And when we go ahead and do that, what we can end up finding is neurons that are seemingly building a map of the image in terms of sort of like, well, what's the most important things to look at? This is sort of a weird sort of type of feature. We find these neurons in the parietal lobe, up here. And this is getting closer to the sensory and motor sorts of areas. And the idea is going to be that we're extracting where are interesting parts of the image, where interesting is a little tricky to find, but something like I'm getting lots of sharp contrasts, I'm getting my feature detectors for faces are working a lot in this area, things like that. And then the idea is going to be that sort of if you build up this map of, okay, I have neurons that are sensitive to different points in this space, and then the activity of the neuron is just sort of, well, how important is this point? So is there interesting stuff at this visual location? Then that sort of map can then guide the visual saccade. So, okay, my eyes are just going to move somewhere. Okay, well, we'll move here next. And then we'll maybe move here, and then move here. So something about, okay, we're extracting out these features, and then we can jump around and do this. Of course, this map is then going to update whenever you move your eyes. Of course, it's now going to change the map, and so on and so forth. Okay. So that seems to be sort of one straightforward thing that, given an image, we might want to pick out where are there complicated features in the image that I might want to look at in more detail. Right. So that seems to be something that the sensory motor system is going to do, is then that information get passed on to control the eye movement. Of course, if this is the case, then we might expect that damage to this area might do some really weird things to attention. And that's indeed exactly what we find. So this is, damage to this area is known as visual or spatial neglect. There's a video there that if you feel like watching it, it just shows a couple of examples of people with this syndrome. The general effect of this is ignoring things, tending not to attend to things on one side of the visual field. So in the standard example, task is I give you this picture and ask you to draw it, or I give you this picture and ask you to draw it, and these are sort of typical responses that you get. I mean, it's not like they're blind to this side of this, like they're still completing the circle, but they're really only paying attention to stuff over here. Again, it's not like we're still getting this line across here, we're still getting, things are there, but we're consistently ignoring stuff on one side of the visual field. Again, fascinating thing that if we were building a cognitive model, it would have to produce this behavior, and wow, that's hard. Cool. One other fascinating little puzzle that the brain has to solve when it's moving the eyes around is, well, hold on, if I'm going to move my eyes around, that's going to massively change all of the data that I'm currently seeing right now. And especially when I've got a situation where I've got neurons that tend to be sensitive to particular parts of the visual field, all of a sudden we're going to massively change what input they're getting. Somehow the brain needs to coordinate that so that it realizes that, okay, look, it's not like things out there in the world are moving really quickly. This is me causing this movement. I'm just moving my eyes or moving my head or moving my neck. All of those sorts of movements are causing the change, not an actual movement in the outside world. So you do tend to find this nifty thing where even in the sensory motor areas that are sort of generating the movement command that's eventually going to go down to the motors, they also sort of send a signal back to the visual system. Basically, it seems to be to try to help the visual system compensate for this change that's about to happen. One particular thing that's going to be happening here is you can get short-term changes in the receptive field of a neuron. What do we mean by that? So normally, a neuron is maybe sensitive to some part of the visual field. So all of for instance, the neurons on the retina are very sensitive, or the ganglion coming out of the retina, very, very sensitive to, oh look, I'm getting input from this particular part of the visual field. And the same is true in V1. We're sort of getting responses to particular parts of the visual field. As you get later in the visual hierarchy, though, this gets to be a little bit more flexible. So for instance, I might have a neuron that is sensitive to apples in this particular area of the visual field, or maybe not apples, so I don't know, red circles in this particular area of the visual field, maybe. And let's say I'm currently looking straight ahead. I'm looking straight at this apple. So maybe, so this is always looking, so this particular neuron is sensitive to something straight ahead of me. I then have, again, saccade, so my eyes start to move. But just before the saccade, what ends up happening, so I'm sending a signal off to my eyes. The eyes are about to start moving. I know from that signal how much my eyes are going to move. And it actually turns out that the receptive field of the neuron shifts about that right same amount. So even though the eyes have not moved yet, but my neuron now starts being sensitive to this part of my visual field. Again, how the heck are we going to have that happen? Starts being sensitive to this part of the visual field, and then the saccade finishes. Now the eyes are pointing straight ahead, and now the receptive field is back to, oh, okay, I'm sensitive to things that are straight ahead of me. This is a little bit weird. This is not a sort of thing that we would typically see in any sort of artificial neural network. This is saying that somehow the weights of, especially if we want to think about a neural network as all of the inputs to the neurons are just weighted sums of their inputs coming in. It's not like the wiring can change. It's not like the physical wiring is going to change to sort of say, okay, now this neuron is connected to these other input neurons. But the system as a whole, with all of these connections, seems to generate this sort of interesting behavior. We actually, one of the early papers out of the lab here was looking at how do you build a network that can do a task like this without having any rewiring. So it's just, here's a fixed set of wiring. How do you extract out the features that allow you to do this sort of shifting, where you're shifting the receptive field without actually moving your eyes or changing your connections. So cool stuff that brain definitely has to do that, again, artificial neural networks generally haven't had to deal with. All right, that's a bunch of weird things about the visual system in mammals, and the hope is to sort of highlight some similarities and differences between the biological systems and the artificial neural networks that we've been talking about so far. As far as projects go, all of these sorts of things, making little models of this sort of thing would fit very well. So in particular, things like, well, there's existing convolutional neural networks that you could train up to do face detection, and then do things like exactly do the remove specific features, remove the eyes, remove the nose, do exactly the experiment that I showed with the monkey, and sort of see how does that affect the performance of the features. Do you get features that change in the same sorts of ways that we saw for the monkey data? Another thing would be to do a convolutional network that tries to build up a salience map, and see what sort of convolutional networks need to do that. There's a few of those out there in the literature, so there's places to start from that way. And again, compare to what the biology is doing, or see how changing different things about that model affect how accurate the salience map is. Cool. All right, so that gives us enough for the comparison needed for talking about the biology, the high-level visual system for assignment 3. So that will be posted soon. And then I think what we're going to go on to do from here is start looking at low-level, and now that we've gone all the way up to a very, very complicated cognitive behavior, we're going to then turn around and go all the way back down and start looking at low-level computational models of individual neurons. Because individual neurons, as we're seeing, the whole brain is doing some really complicated stuff, but it's also the case that individual neurons are also doing complicated stuff that we also might want to build computational models of. So we'll see how that goes. Thank you very much, and talk to you later.